I'm Greg Bowman, and I'm an associate professor at the Washington University School of Medicine and the director of the Folding at Home project. And I'm really delighted to join you today virtually to tell you some about how we've been using a massive distributed computing project called Folding at Home to understand and combat COVID-19 disease. So to give a little bit of background, the Folding at Home community at large and my research lab in particular are very interested in understanding how these molecular machines called proteins work. And this is really important because proteins are responsible for everything from our ability to contract our muscles, to sensing light in the eye for vision, and basically any other active task that you associate with life. They're also important components of infectious diseases like bacteria and viruses. And when human proteins malfunction, this gives rise to diseases like cancer and Alzheimer's disease. And so you can imagine there's myriad ways we might want to shut off particular proteins to prevent diseases like cancer or viral infections, or to improve the function of other proteins to counteract disease. And one of the challenges here is that these molecular machines called proteins, like the machines that we're used to working on macroscopic scales like cars, have lots of moving parts, but they're very small. And so experimentally, we're often limited to getting snapshots of what these proteins typically look like this one. So this is a protein that we've done a lot with called Betalactamase. And what we're looking at here is a representation where each sphere represents an atom in this protein. And they're colored according to what element they are. So carbon is in gray, nitrogen in blue, oxygen in red, for example. And we can learn a lot from this single snapshot. So for example, there's this pocket here that we call the active site, and that happens to be where this protein performs its function. And in this case, that's to bind to antibiotics like penicillin and catalyze the degradation of these compounds. And so these proteins are actually expressed by bacteria to defend themselves from our antibiotics. And as you can imagine, there's great interest in designing inhibitors of these proteins to prevent them from performing this function so that our antibiotics continue to work. And snapshots like this have provided a lot of information, but this protein is likely full of moving parts that we just can't see by zooming in on it with some magical microscope that doesn't exist. And so it turns out that computer simulations are an extremely powerful way to get at these moving parts because we can track how every atom in this protein moves over time. And so I want to show you just a snippet of one of these simulations to give you a sense of what that might look like. And in particular, I want to draw your attention to this region that I've highlighted with this label here, where a larger change in the protein structure will occur. And so here you can start to see all the atoms wiggling and jiggling around, and it's pretty subtle at first, but right about now you can start to see this larger opening form in the protein. And this is really exciting to us, and we call it a cryptic pocket because we wouldn't have guessed it was there from that initial snapshot. But we're starting to understand how the formation of this pocket might actually contribute to the function of this protein, and definitely provides us with new opportunities to design drugs that inhibit this protein. Because they can bind in this, we've come up with molecules that can bind in this pocket that we, again, didn't know existed before, and actually shut this protein off. And so we're very excited about new opportunities like that and their application in numerous diseases. And so to get at this is really valuable, but it's also really difficult because running an individual simulation that's long enough to get at these sorts of processes that we're interested in can easily take hundreds to millions of years on a single desktop computer. And so one of the things that my group and other groups in the Folding at Home community have worked on is methods that allow us to break these enormous simulations up into lots of smaller bits so that we can run them in parallel on independent computers. And taking this idea to its logical extreme, we as a community realized, well, we should be going out on the internet and inviting anyone with a computer and an internet connection to help us run these simulations. And thus the Folding at Home project was born about 20 years ago. And so this is just a map of the globe showing a little pinprick of light where everyone who's currently participating in the project is currently. And so this has provided some really tremendous compute resources. At the beginning of 2020, we had about 30,000 devices around the world helping to run simulations and advance our work on diseases like antibiotic resistance and cancer and Alzheimer's disease, among others. And just to give you one more example, this is a protein from Ebola virus that my group was very interested in. So here, instead of showing you every atom in the protein, I'm actually showing you what's called a ribbon diagram. And this just traces the chemical components of the protein. So it actually consists of a linear chain of chemicals called amino acids. And so this ribbon just traces that chain and shows how it's folded up on itself to form the three-dimensional structure of the protein. And if you're interested in some time, I'll be happy to show you the all-atom representation like I showed you with beta-lactamase before. But what was very interesting to us about this particular protein is that it plays an essential role in Ebola's replication cycle. But unlike beta-lactamase, it doesn't have any nice deep pockets where a small molecule drug can likely bind and shut off the function of this protein. And so in this case, we were very interested in finding cryptic pockets because this would be the only way to potentially target this protein. And indeed, through our work, we found this dramatic motion that opens up this pocket here. And we've subsequently shown through a variety of experiments that this pocket is real and provides us with a handle to actually shut off the function of this protein and potentially combat Ebola virus infections. And so given this background, you can probably imagine that once it became clear to us that COVID-19 was going to become a pandemic that affected everyone around the globe, it was clear how we could pivot quickly and start applying all of these tools to, instead of tackling Ebola and bacterial infections, for example, to understand how the SARS-CoV-2 virus that is responsible for COVID-19 disease works, and to identify therapeutic opportunities like these cryptic pockets and aid in the design of new therapeutics. And this was a really amazing experience. Obviously the pandemic has been very bad and I'd rather it had not happened. But one little bit of silver lining was that it was really exciting to see people come together and contribute whatever they could to help combat the pandemic. And so in the case of Folding at Home, we went from having these 30,000 devices around the world contributing to our efforts to having well over 1 million devices within a matter of months. So here I'm showing the downloads of our Folding at Home software over time. And you can see that within the first couple months of the pandemic, in blue here, we quickly grew to over 1 million new downloads. And this kind of tracked with the growth of the pandemic and people's growing awareness that none of us was going to be unaffected by this pandemic. And so the upshot of this is that we took this extremely powerful computational resource and turned it into the most powerful supercomputer in the world. So here I'm showing a measure of performance computing in terms of exaflops or floating point operations per second, and showing this for the 10 largest supercomputers in the United States, and then Folding at Home. And so what you can see is that Folding at Home is the first computer to break this exascale barrier. And with a very conservative estimate of its performance, actually was giving us over 5 times the performance of the fastest supercomputer in the United States, the Summit supercomputer. And so this opened up enormous opportunities for our research team to make progress on this very important problem of understanding how this virus works and helping to devise ways to combat it. And so coming back to this all too familiar version of image of the virus, one of the first things that captured our attention was this set of 3 proteins called the spike complex on the surface of the protein. So each of these red protrusions is a copy of the spike protein. And this is really interesting because the spike is the first point of contact between the virus and a potential host cell. And so its job is to bind to a protein on the surface of a human cell, for example, called ACE2, and actually initiate infection of that cell with the virus. So it has this really essential role in the virus's replication cycle. And it also is an interesting vulnerability because being exposed on the virus's surface like this, it's an obvious target for components of our immune system to try and recognize and then trigger an immune response to quickly clear a new infection. And so the SARS-CoV-2 virus has actually evolved a number of defense mechanisms. And one of the ones that really interested us is called conformational masking. And so the idea is that instead of presenting this key interface of the spike all the time, so that any time the virus can interacts with a host cell, it can potentially bind and initiate infection, this complex of proteins actually tends to fold up on itself and protect this key vulnerability, and then has to open up some of the time to kind of feel out if there's a potential binding partner there. And so what we're seeing in this image is actually this closed up, protected form of the protein that's more difficult for our immune systems to detect. And so what we wanted to do is see if we could actually capture the opening of the spike and get a sense of what that looks like and what vulnerabilities it might present that we could take advantage of in the development of new antiviral drugs or other therapeutics. So here I'm showing you an alternative representation of the spike. Again, I said this is made out of 3 proteins. So each of these 3 ribbons in different colors is one of those proteins. And the transparent surface is a space-filling representation, just to remind you that there isn't a lot of open space in this complex. It's actually a quite dense glob of atoms. So up here at the upper left is where the spike is attached to the surface of the virus. And down here at the lower right is where the protein has the capacity to bind to a host cell when it opens up. And so I'll start this movie, and you can see that at the beginning, like beta-lactamase, this wiggles around some. And that's about all that anyone else has been able to capture with computer simulations, because this is a really big protein that undergoes slow motions. But with the compute power of folding at home, we were actually able to capture this dramatic opening motion you can see here that exposes the surface of the virus that actually binds to ACE2 and is the key piece of this protein that initiates infection of a host cell. And this was really exciting to us because it's just such a computationally difficult problem and has really interesting implications for our understanding of the SARS-CoV-2 virus and related coronaviruses. So again, we do have this massive computational resource. So beyond just simulating the spike from SARS-CoV-2, we actually went and simulated the spikes of the original SARS virus, SARS-CoV-1, and another coronavirus called NL63 to compare the different behavior of these spikes and understand what implications that might have for the different lethality and ability of these viruses to spread. So what I'm showing here is the probability that the different spike complexes from these viruses adopt structures that are open to different extents. And so here's some representative structures showing that to the left are these very closed structures, and to the right are these highly open structures that can actually bind ACE2 and trigger infection. And so what we see is super interesting, where the original SARS virus has the highest probability of being really open. And this has caught our attention because that virus was far more lethal than SARS-CoV-2, but actually failed to become a global pandemic. In contrast, NL63 over here has the highest probability of adopting very closed conformations. And this is interesting because this virus is actually associated with symptoms more like the common cold and has spread quite broadly, but not caused the sorts of problems that the SARS-CoV-2 virus has. And right here in the middle in black is the SARS-CoV-2 virus. And so what we think it's doing is actually striking this balance between adopting closed conformations where it can hide from our immune systems and open conformations where it can actually bind to our host cells, and initiate infection to be better able to spread readily than the SARS-CoV-1 virus, while also hiding from our immune systems better than that virus and achieving the ability to spread rapidly, as we've seen in the last 8 months. So beyond this understanding, this also gives us a lot of information about how to target this protein. So for example, from just the closed structure, you'd think that the only way to target this protein would be to target the surface, which is actually protected by other mechanisms that I won't go into now. But from having these structures of these highly open conformations, we can actually see that there's all these other surfaces that we could potentially target that we wouldn't have guessed from knowledge of just this closed structure. And these surfaces are actually vulnerable, and so they're actually far more attractive targets than the surface that we see in these closed structures. And indeed, there's already an antibody that people have developed that binds to one of these surfaces that we uniquely capture in our simulations of this protein that people previously couldn't quite understand how it actually bound to the spike and protected our cells by preventing the spike from engaging with ACE2. And so we're hoping that we and others can now start using this structural information to inform the design of other therapeutics. And so we've made all of the data publicly available online for anyone who wants to join us in mining it for these opportunities and guiding antiviral efforts. So like I said, we do have the biggest supercomputer in the world. And so in addition to doing these simulations of these different spike complexes, we've also run simulations of every possible protein we can, covering most of the proteins from the SARS-CoV-2 virus. So just to give another example, this is a protein from the virus called the main protease that also plays an essential role in the replication cycle of the virus and has been one of the very primary targets for efforts to design antivirals. And so like Betalactamase, this is an enzyme that catalyzes a chemical reaction. And I won't go into the details, but we'll just note that the active site where this reaction occurs is in the upper left of the protein here. And so that's been the main site that the drug design community has been focusing on. But through our simulations, we actually found all kinds of motions, as in the spike, where this protein opens up and creates these unexpected binding pockets that we've been finding are very appealing targets for drug design campaigns. And so we've actually found 2 of these pockets in the main protease, as well as many as 50 cryptic pockets in other parts of the SARS-CoV-2 virus. So this is a real wealth of structural information that we and we hope others will be using to inform the design, again, of small molecule drugs and antibodies and other therapeutic strategies. So I won't get to go too much into the detail about how we're doing this, but here I'll just show you another snippet of a simulation, again of the main protease, but now with a surface representation of the protein instead of this ribbon diagram. And here I'm showing a potential small molecule inhibitor bound in the active site of that protein. And so what we've been doing in collaboration with the COVID Moonshot, and this collaboration, I should note, is spearheaded by John Kadera at Memorial Sloan Kettering and Vince Wills at Temple University, is helping to screen libraries of tens of thousands of potential small molecule inhibitors of this protein with folding at home. So we're running simulations of these small molecules with the protein. And from that, we can calculate how tightly they bind to the protein. And so now, instead of having to spend a lot of person hours and money and other resources to make all of these compounds, we can computationally determine which are most likely to be potent inhibitors of this protein, and then focus experimental efforts through other groups in the collaboration on those small molecules. And so this has been going superbly, and the effort now has really exciting small molecule inhibitors of the protease that are going into animal testing. And so I hope this will position us well to combat SARS-CoV-2 and more likely than not, future outbreaks of other coronaviruses that are likely to have very similar proteases. So I hope that gives you a sense of what we've been doing with folding at home. With that, I'd like to thank everyone on the research team that's really dropped everything else and put a lot of hours into pushing on this research. Of course, I'd like to thank everyone in the folding at home community from across the globe that's contributed their computing power. And this includes both individual citizen scientists and a number of organizations. So Pure Storage has generously donated us over a petabyte of storage space to store all of the data that we've been generating, which is in the hundreds of terabytes already. Microsoft, AWS, Oracle, and Cisco have helped us to get infrastructure up and running on the cloud so that we can productively harness the computational power of the hundreds of thousands of people that are contributing their compute power. And companies like Avast have helped to support extra software developers so that we can keep up with everything that's going on. And I'd also like to thank all the funding sources that made this possible. And I hope that you'll join us and contribute your compute power to Folding at Home and help us to keep moving this really important research forward. So please take a look at our website at foldingathome.org. Thank you very much.